Right, so what we're looking at is question 9. And this is normally in paper 2, so this is the more difficult version. So you've got to read all this. Venus flytraps carry out a process of photosynthesis, and miraculously, I'll go to the end. The leaf trap now serves as a digestive organ to dissolve the soft tissues and cell membranes of the food. It produces acid and enzymes. The insect body is broken down over a period of 5 to 10 days, and the products of the digestion are absorbed. Such a good reader. Right. You have to read that, or sometimes it's good to read that, but I've seen that you don't really need to. Just follow this. Right, so let's have a look. They want us to know what the process that releases energy from glucose is called what? Right. So the process that releases energy from the from glucose is called respiration. Right. Remember, respiration is not the same as breathing. Okay, that's a common mistake. Breathing is more to do with ventilation, where air moves in and out of the lungs. So remember that. Okay. Right. A carnivore eats. Carnivores eat animals, or yeah, animals. Herbivore eats plants. Carnivores plants attract insects for food. Give a reason why other plants need to attract insects. It's for pollination. So then they can take the pollen to other plants, or it can bring other pollen to the what's the word? Is it stamen? No, to the stigma. I always forget that stigma. Okay, there you go. The Venus flytrap can be placed in at 2 different trophic levels. Name these. So it can be a producer because it makes its own food as well as it grows. Producer, and it's also a consumer. So it's a what would it be? It could be a yeah, just consumer. So let's have a look at that. Right, so they don't want you just to write the word consumer. So it's a secondary consumer or even a tertiary. 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 Let me get the spelling right. Hello. No. Why it's not deleting? Let's try that again. Tertiary. Secondary or tertiary consumer. Right. The trap closes if 2 of these hairs are brushed in quick succession, or 1 hair is touched twice. Why is this an advantage? So what happens is it stops the plant wasting energy. It stops the plant wasting energy by closing unnecessarily or accidentally. Yeah, so then it's just wasting energy. It's of no benefit. Okay, so let's go down. Explain how the changes in concentration of solution in cells can lead to water entering cells. Right, entering cells. What they're saying in line 23. right, concentration of solution, so entering the cells, right. So you've got a cell, you want water to solution to enter, so that means this is low concentration and this is high concentration. So we can say the solution enters the cell by diffusion from an area of high concentration to an area of low concentration. Okay, so that's good. Right. Oh, okay. Look, I missed a keyword there. So solution entered by diffusion. Right. Because it's water, I would not use that word. You use the word osmosis. So the solution enters the cell by osmosis from an area of high concentration or high. Right, let's leave this on. It's good actually. Water potential. Let me write this word here. Potential. To an area of low, not concentration, but low water potential. That's it. Right. I thought it was just diffusion, but it's water. And when it comes to water, you need to use the word osmosis. And it's better to use an area of high water potential to an area of low water potential. Good. Right. Okay. Let's move on. Right. Question 10. Right. Lipase is an enzyme that digests lipids into fatty acids and glycerol. Name the part of the digestive system where lipase is released. Pancreas. Whoa, pancreas. Pancreas. Right, in terms of digestion, it releases lipase, protease, but the one, okay, look, watch this, protease, and the one that it releases is trypsin. And then you've got lipase, and then you've got amylase. Right, for digestion, we're not interested in insulin or glucagon. Right, a drug X that inhibits means prevents or stops. Lipase is used to help people lose weight. So what would happen is, look, your body cannot absorb lipids directly. It has to break them down into glycerol and fatty acids. Right. So it has to break it down into glycerol and fatty acids. If this drug X stops lipids becoming glycerol and fatty acids, what will happen is the lipid will not get used by your body and it will leave your body with the rest of the poo or the feces. Right. And that's what it does. So what it does, it stops. So let's explain how. Let's go over here and explain how with a red pen. It prevents lipids being digested or broken down into glycerol and fatty acids. right, and being absorbed into the blood and becoming part of your assimilating into your body, becoming part of cell. Right, okay, so you've got a set up here, pH meter. pH measures the how acidic something is. It measures acidity. Right. So they carry out this. Explain which temperature of water should be used during the investigation. Right. Human beings are at about 37 degrees, so they should use 37 degrees because this is the optimum temperature for enzymes because that's our body temperature. So there's two things here, right? We've done 37 degrees and we've used the word optimum for the enzymes. And that's where you're going to get your 2 marks from. So remember the optimum temperature or the optimum pH or the optimum whatever it is, yeah? Okay, right, let's move on. Right, vitamin A, it helps with eyesight. It stops something called night blindness and then eventually total blindness. It's also got a function with skin. So, but vitamin C helps with eyesight. It helps with vision. Right, that's vision. Vitamin C is to do with the skin. It helps join the layers of skin. Basically, it prevents scurvy. Vitamin T is for both bone growth. Iron is for red blood cells, right? What in particular? It is used in the making of hemoglobin. Right, and what does that do? That helps carry oxygen around the body. For what? For aerobic respiration. What did we say aerobic was? It's with oxygen. What's the opposite of that? It's anaerobic. That means with oxygen. Right, dietary fiber helps with peristalsis. This is movement of the food through the gut. It helps keep it clean. Right. So what I want to do is now, okay, a test for glucose. So what you do is you add Benedict's solution. You add Benedict's solution. You heat in a water bath, and depending on the amount of glucose it contains, the solution goes from blue to brick red, and that's what happens. Right, okay, let's keep moving on. The meal also contains lipids and proteins. Give 3 chemicals found in lipids. So you've got carbon, hydrogen, and oxygen. Right. 2 substances involved in digestion. Naming the organ that produces. So bile, we know, is produced by the liver, and lipase is produced by the pancreas. Okay, bile. Right, remember we said bile is not an enzyme, but it emulsifies large fat globules. Emulsifies. It turns into big blobs, so increases the surface area increases, so the enzymes can act on it and digest it. Right, okay. Number 12. Right, so you've got teeth, esophagus, stomach, large intestine, small intestine, rectum, and pancreas. Complete the table. So you use to chew the food with your teeth. Has most villi for absorption, so it's the small intestine. It's normally the second part of the intestine, and that's called the ileum. Produces hydrochloric acid, and we know that is the stomach or stomach. Stores feces, the rectum. So remember what we said, right? You've got your small intestine, you've got your large intestines, it goes up, down, and round. So we said the first part of the small intestine is called the duodenum. The second part is called the ileum. The large intestine, so that's the small intestine. The large intestine, this part is called the colon. It absorbs water, and this part is called the rectum. It stores the feces and it's expelled out of the anus. So all these words you've got to know. Right, let's move on. We're on 17, 18 minutes. Right. How does the liver help digestion? Look at this posh word, alimentary, alimentary, medullary canal. Wow. So that's the small intestine. Or the intestine. Okay. So what does the liver produce? The liver produces bile. What does the bile do? The bile does 2 things. It emulsifies the lipids. So the enzymes. So right. Okay. Whoa. Let's just go back. Let's put this in. Emulsifies the lipids to increase surface area for enzymes to act on. Enzymes to act on. Right. Right. It also neutralizes. So it neutralizes the acidic content from the stomach. Right. So it is alkali. Right. Let me just quickly explain it. So you've got the liver, you've got the gallbladder, you've got the bile duct, and here is the stomach, and here is the small intestine. So what happens? You've got the gallbladder, Okay, the gallbladder, that's the liver. So the liver produces bile. It's stored in the gallbladder. It travels down the bile duct into the small, so this here is the small intestine. Small intestine. So what comes out of the stomach is partly digested food and it's acidic. So bile has to be alkaline. Let me write that properly. Alkaline. And when it mixes here, it neutralizes it because the small intestine can't handle it. Right, here you go. We need to draw a plant cell, leaf cell. So they've given us the cell wall. What I would do is draw a self membrane, cell. So that there is the cell membrane. I would draw a permanent vacuole. Permanent vacuole. I would draw a chloroplast. You always have cytoplasm in there. Cytoplasm. You've got ribosomes, and you've got mito. That's terrible. That. Mitochondria. Okay, there you go. Right. You've got to learn about bacteria cells as well. So you've got rabbits cannot digest cellulose. Oh, I thought they could. Bacteria living in the large intestine of rabbits can digest the glucose. Okay, yes. I get it. Didn't make sense for a second. The rabbits eat what? Really? Rabbits eat their own feces to obtain this glucose. Ooh. Okay. That's good job. We're not eating. Give two differences between the structure of bacteria and plant. Right. Look, this is a bacteria cell. Bacteria cell has a cell wall, so that's not different. But bacteria cell has something called a buccal. It's a tail, but they call it flagellum or flagella. Flagella. Right. Plant cell has a real nucleus, but they have plasmids. Bacteria have plasmids and a nucleoid. So this is the DNA. Nucleoid. Okay. Yeah, there. So it has a flagellum and it has a plasmid. So this is a prokaryote or karyotic cell because it does not have a nucleus. Eukaryotic cells have a nucleus. Prokaryotic cells have no nucleus. Eukaryotic cells have a nucleus. Right, describe a test for glucose. Okay, so what did we say? Add Benedict's solution, heat in a water bath, and once you've done that, what do? It will turn brick red. Don't just put red, brick red if glucose is present. So you have to write everything. Oh, right, okay, that's it. 25-minute video. Right, okay, that's done.